Well, good afternoon, everyone. Thanks for joining us for our second webinar. We're just getting a few more people connected. So I want to give about 30 more seconds if you'd like to participate in our introductory poll. Navigate to the web address at the top of the slide, and you can share a little bit about yourself. We are recording this webinar, and we will post it on the National Sea Grant Law Center's website following today's event. All right, well, I'm going to start recording and we're going to get started. Okay. All right. So hi, so I'm Stephanie Otts. I'm the director of the National Sea Grant Law Center. I want to thank you so much for joining us for our second installment of the NOAA Sea Grant Water Resources Visioning Teams 2021 webinar series. That was a mouthful. So we're a professional network of Sea Grant personnel working to address water-related challenges. If you are staff of a Sea Grant program and would like to join the Water Resources Visioning team, all you need to do is send me an email and I will get you on our distribution list. We welcome everyone, and we would love to have as many people who are working on these issues as possible. So in 2021, the Water Resources Visioning team launched a webinar series to raise awareness of the team's efforts in this area and to highlight innovative research, outreach, and education activities of the National Sea Grant Network. And we kicked off our webinar series in January with the focus on COVID-19 in the water environment. If you missed that webinar and you are interested, a recording is available on the National Sea Grant Law Center's webpage for viewing at any time. For our second webinar today, we chose to focus on contaminants of emerging concern with special attention paid to microplastics and Pfas, Pfoa. Congressional attention on these contaminants has been growing, and we know that there is strong Sea Grant work to build off as federal funding or any other opportunities emerge. And so we wanted to provide this opportunity to start the conversation and learning. So before I start, I wanted to thank the members of our planning committee for all their hard work in making this webinar happen. As all you will know, it always takes many hands to make these things work. And so really appreciate their efforts. And you'll be hearing from many of our planning committee members today throughout the webinar. Logistical and planning support for the webinar series are provided by the National Sea Grant Law Center and Karen Bareford, the National Water Extension Liaison at the National Water Center. So it really takes a village to pull off these efforts. And with that, I'd like to introduce our first speaker. So Steve Jones is a member of the Water Resources Visioning team, and he is Associate Director of New Hampshire Sea Grant. And he's going to kick us off by kind of setting the stage for our conversation.
Okay, thank you, Stephanie. Great to have you all here. What I'm going to be doing is trying to, in 5 minutes, give you an overview of CECs. This is a very complex issue and it's changing all the time. And so the next slide, will I be asking you, Stephanie? Okay. I'll really be giving a sort of a general framing of the CEC issue. I want to touch on the fact that there are national assessment approaches, taking a look at CECs that are present in coastal aquatic ecosystems, as well as give an example of a study in New England that New Hampshire Sea Grant is funding, looking at wastewater treatment facilities as a source of CECs to coastal waters. Next.
Great. Yeah, and so next we just had a poll question just to kick us off. And if you could navigate to that web address at the top of the screen and just type in a quick example that jumps to your mind about a CEC. We'll just give it about 30 seconds or so for people to.
And by way of this, they'll answer the question that I'm posing in this slide. So that's good.
We also like this word cloud function because that means at the end of the webinar, all of the participants has helped us create an image that we can use for our advertising and other kind of efforts to figure out what the hottest topics are. So this is great. All right. We'll just give it about 5 more seconds. So if you have a burning thought, type it in fast, and then we'll keep going. Okay, go ahead, Steve.
Okay, so what are contaminants of emerging concern? They're a complex group. If you want to make a list, the EU tried to do this a few years ago. I know they're probably moved on from this, but there was a study where they came up with a list of over 2,700 individual and groups of substances. So it is a wide-ranging topic. In this table on the right, we have classes and examples of CECs. And this includes a number of chemicals that you can see, the alkylphenols down to the sterols and examples of those. But it also includes microbes, microplastics, nanomaterials, and harmful algal blooms. And so this topic is very wide ranging. And of course, on the left, there's the infographic showing that most of these materials are land-based. They find their way into our coastal waters. And from there, their fate, they may disappear, or they may be taken up or cause effects on the ecosystem or into human health. Next slide. So what do we know about contaminants of emerging concern? I'm not going to answer a lot of these questions. I'm mostly posing the questions that we're all asking ourselves. But we do know that contaminants of emerging concerns are not well regulated. For example, in the toxic chemical realm, there are toxic pollutant and priority pollutant lists that are quite outdated, but they're what we have and are still used by EPA states and authorized tribes to try to develop water quality criteria. So, but overall, these are contaminants of emerging concern because they're not really in the regulatory framework yet, or they're emerging into that. There's been a lot of recent work in the field of CECs, including research on monitoring technology, analytical chemistry, exposure effects in humans and ecosystems, as well as remediation. But overall, our understanding is still quite in the nascent stage in terms of how well we understand it. So the next slide. So how do we determine which are of most concern? Well, obviously, Stephanie mentioned microplastics and Pfas compounds, perfluorinated compounds. Those have emerged into contaminants of concern. But other factors include sources. Are there large volumes? Are there high concentrations? Are there ubiquitous sources that we have to deal with? What is their fate? Are they persistent? If they disappear rapidly after they hit the coastal waters, they're not a concern. But if they persist or if they're compartmentalized into the ecosystem, that's a concern. And their effects, first of all, if they're toxic, that's going to be a concern if they have impacts to ecosystem health. But is there a pathway for exposure is another key factor as to whether or not we concentrate on certain compounds or not. Next slide. So I do want to say that there's a lot of national assessments taking a look at the CECs in the United States, federal agencies, state agencies, NGOs, academic institutions. But what I want to do in my short time is to take a look at one, and that's the NOAA INCOS Muscle Watch program, and take a look at what they've been doing over the last decade. The next slide. So this is a map on the left of most of the United States. This highlights some results that were from 4 studies in the Gulf of Mexico, Charleston Harbor, Chesapeake Bay, and the Gulf of Maine that Mary Ryder and Dennis Apetti presented at CTAC a few years ago. And you can see on the right the list of 254 target compounds. Other agencies may use a larger list or a smaller list. So there's a whole realm of different compounds that were looked at. And the one piece I'll just share with you in this short time period I have is this graph on the left, this blue strung out bar graph where we see that only 7% of the 254 target compounds were detected in more than 10% of the samples from all combined in all these areas. And so a lot of the compounds that we're looking at may not be present, or may be present scattered or in a rare fashion. Next slide, please. Just to hone in on the Gulf of Maine project, which I was involved with, where they partnered up with our existing Gulf Watch program, which was started in 1991 and really mimicked the Mussel Watch program. So we were very compatible in terms of what we were looking at. And there's a report that will be coming out any day now about the contaminants of emerging concern in the Gulf of Maine. So the next slide just shows a little bit of the results. And again, just touching on things briefly, these are distribution maps showing presence in blue or gray, absence of compounds. And you can see PFCs on the left and pharmaceutical and personal care products on the right. And you can see that a lot of these compounds were not detected. They're detected rarely. The x-axis in each one of these graphs are the sites that we used. Up at the top, you can see that's Massachusetts, New Hampshire, Maine, or Nova Scotia. We didn't find much up there in Nova Scotia, but one of the things that came out of this is the most frequently detected compound was DEET, an insect repellent, found in 88% of the sites that we looked at. So this tells us something. Which compounds are the ones that we need to take a look at? And what are the impacts of DEET on the environment? So it goes both ways. So we're getting a feel for what to look at and what's of most concern. Next slide. And I'll leave you with just a brief mention of what New Hampshire Sea Grant is supporting. Doctors Paula Mauser and Jim Malley and their students have been looking at PFAS moving through wastewater treatment facilities using different treatment approaches. And the next slide just shows a little bit of their results where they were measuring what was coming in and measuring what was coming out. It didn't really make sense. They started looking at the solids, and they're finding that the solids in the wastewater treatment facilities contain more PFAS species than the aqueous phase, and they're finding some compounds in the solids that they didn't detect in the influent. So a lot of questions are in your mind probably with just that mentioned, but I'll leave that there and let others get into more detail. So the last slide, just want to say that what can Sea Grant do? We're already involved through this webinar. And I think we all agree that the next step is to define a strategic role for Sea Grant in addressing CECs. Thank you.
Great. Thanks, Steve. Thanks so much. So we also have another kind of part of the kind of setting the stage. So I know you probably have questions for Steve, but we have another poll question, and then we're going to have kind of a follow-up presentation to continue setting the stage by Mike Mezzacapo, a water resource outreach specialist with Hawaii Sea Grant. He also serves as the 2020-2021 Lake Champlain Sea Grant affiliate. So we want to take about a minute to give the audience the chance to engage in this polling question, and then I'm going to stop the screen share and Mike is going to have a few remarks. Great. Okay, Karen, it looks like the bars have stopped moving. So give you a couple of seconds to capture it, and then I'm going to stop the screen share.
Thank you. I've got it.
Got it. Okay. Okay. Okay, go ahead, Mike.
Thanks, Stephanie. I appreciate the opportunity to speak today. This topic is a passion of mine as I used to work in healthcare. I don't have any slides today. I chose to do a fireside chat. So I'll speak quickly about the connectivity between Sea Grant programs, opportunities for collaboration, and give some examples of how integrated CECs are into our life and how Sea Grant could possibly play a role in addressing CEC problems. So it's clear just how many potential CECs there are from Steve's talk, but I wanted to reiterate that CECs impact the people and environments in every state and region served by Sea Grant programs. Many CECs individually or occurring in the environment as mixtures. They have unknown human health effects, ecological impacts, life cycles, toxicity levels. To understand how widespread the problem is and working off Steve's presentation, I think it's important to highlight the many sources of CEC transport mechanisms that exist widely across our programs, right? So these include industrial processes or byproducts, wastewater discharges, land-based application, leachate, stormwater runoff, mismanaged waste streams, and food webs. CECs cross-cut many, if not all, of our Sea Grant focus areas. They impact fisheries and aquaculture, water quality, tourism, and the economy, among others. Similar as well as different types of CECs affect our collective communities. Some chemical compounds are broadly used no matter the geographic region, while others, and they're spread via consumer products or waste transport, while others are really specific to regions that are specializing, say, in certain sectors like chemical manufacturing, drilling, mining, or refining. The Sea Grant network understands the dangers of CECs and has a strong foundation in addressing some of these pollutants, especially with respect to outreach, prevention, and monitoring. For example, Illinois, Indiana, and Wisconsin Sea Grants hire dedicated specialists to address things like CECs. Programs like Oregon Sea Grant instituted a drug take-back program to keep medicines out of the environment, and the Water Resources Visioning team is holding this webinar. The opportunities are limitless to increase our formal communication and share resources across Sea Grant programs. This includes leveraging different respective programs and their specialties on behalf of their local coastal communities, but also nationally. Despite the opportunities available, though, there are some gaps Sea Grant programs could fill, including doing more to connect research to policy and deepening outreach and engagement efforts with stakeholders that really lead to behavioral changes. This does require transdisciplinary work across physical and social sciences. Think chemistry, economics, ecology, toxicology. And lastly, more work is needed in the social science realm to address and emphasize environmental justice impacts from CECs. Many communities of color are adjacent to pollution sources that emit CECs, and they bear disproportionate or unknown health impacts. Examples of locations could be ports, landfills, chemical plants, farms, or incinerators. In addition to thinking about where we can really step up our efforts, we need to envision how to make an impact. So though our geopolitical boundaries separate Sea Grant programs, ecosystems are often intricately connected. I think an example comes to mind, the Great Lakes region. There are 8 Sea Grant programs that border the lakes, each have their own universities, state laws, regulations, and conduct. However, the use of a CEC in one area will likely impact a broader area and possibly the entire system and beyond. So thinking about these problems more broadly may be beneficial to help crafting holistic solutions. We need to recognize the impacts of aquatic health can also vary by location and may or may not be detected depending on levels of development in an area or certain species nearby. And smaller communities, both human and ecological, can disproportionately be impacted by larger communities upstream. So to help set the stage for our speakers as well as our panel discussion later, I'll just touch on a couple of personal examples of challenges that are faced by consumers across programs. Due to the time, I can't give any exhaustive examples, just know there's a lot of good ongoing work across the Sea Grant network that I didn't have time to highlight. So I've lived in both New England and Hawaii, and recognizing that many of us probably have CECs in our homes, whether we know about them or not. And proper disposal seems to be one of those hurdles that is hard to overcome to prevent these products from entering the environment. Early intervention by encouraging consumers and organizations to only purchase or dispense what they need. For example, doctors could only prescribe a certain amount of medication rather than giving a month's supply. So by doing things like that, we might be able to reduce the amount of these products from even entering the environment or waste streams. And one way Sea Grant programs really could be impactful is to assist communities with more information and outreach efforts for proper disposal and dispensing of these products. As an example, in Vermont and New Hampshire, which I've both lived in, there are limited take-back days, we call them, often only one a year for household waste products. So paint, pesticides, cleaners, maybe bottles of insecticide with DEET in it, making disposal a really critical barrier. Products often end up sitting in your garage and then end up being disposed in a landfill or sometimes dumped onto the environment. And one stat from nearby New York indicated that there was about 100,000 tons of these products that end up in trash cans every year. So by expanding donation options or paying to take products back that may cause harm to the environment, like the like bottle deposits or can deposit return programs, it could be one way to really lower that hurdle and capture products before they enter the environment. So in Hawaii, there are also similar challenges that are faced in disposal of these products. I had major surgery last year and I had a lot of medications left over, right? I could only find one place to dispose of the medicines on the entire island of Oahu, and it was often full. The box, there was just a little box that someone came and picked up and it was full, so you couldn't even get rid of it. There's only one trash landfill for construction waste on Oahu, and all the other waste is incinerated. So meaning potentially hazardous items that are thrown away could be emitted as air pollution. So some of these wastes also have to be transported back to the mainland, which increases the potential for spills during transport. But there are successes. So Hawaii developed solutions to reduce potential CECs from entering nearshore waters through its ban of certain sunscreens with oxybenzone and octinoxate by engaging the main contributors, which were tourists, in positive actions. And that allowed Hawaii to better protect its own aquatic resources, but also get consumers and visitors to really think about the products that they were using and how they entered the environment. So my homework before I leave here is just to get you to think about how we could all collaborate in our respective programs, maybe in ways that we didn't envision before, so that we could tackle this broad-reaching issue. Thanks.
Great. Thanks, Mike. Thanks so much. So really appreciate that. So I think we have one question, which Karen can ask as I start to get the screen share back up for our second speaker. But just remember for everybody that you can ask questions in the Q&A box for any of our speakers and later our panelists.
Thank you, Steph. Okay, so we have a question from Peter, and he wanted to know if possibly why the neonicotinoids, and I probably didn't pronounce that right, but are missing from this list since they are used to coat most of the corn, soybean, and wheat seeds that are used in millions of acres of farmland. Stephen or Michael, could one of you address that by chance?
I think I kind of addressed that in what I was talking about. Just there's lists and but there's no one list. There's a lot of compounds that are emerging as contaminants that are not making outdated lists, and there's new lists that are not including it. It's kind of, my view of this is it's not very well organized, and so the list that I had did not include that. It was not included. Those were not also included in the Muscle Watch array of analytes either. So good point, but I tried to find the ultimate list and I couldn't find it.
I don't know that there ever will be, right, an ultimate list. I mean, just because we keep adding chemicals, that are added to different products that we use, or we're finally learning about new studies that discovered, one lasts longer than we thought. So really, I think it also stems from kind of how we approve chemicals for use as a national policy, and that might kind of just continue to add to our list, and we'll kind of almost never catch up unless we change something in terms of how we approve these items.
Good. Great. Well, thanks, Steve and Mike, so much. They'll be back later in the webinar for the panel, but now I'd like to introduce our next speaker. So Carolyn Foley is the research coordinator with Illinois Indiana Sea Grant, and she's going to be talking about microplastics. Take it away, Carolyn.
Thanks, Steph. And thanks to everybody for giving me some space to talk about some research we've done on microplastics in aquatic organisms. If you could click, please. Okay, so just to be sure we're on the same page, microplastic pollution, it's less than 5 millimeters in size, so pretty tiny. It can come from a whole bunch of different sources, and we have good evidence that it is absolutely everywhere. It's in open waters, it's in tributaries, it's in the sediment, it's in the air, it's in the oceans, it's in the lakes. It's absolutely everywhere. So if you could click again, please. So we also have evidence that it is being consumed by aquatic organisms. This is one example of a study from Lake Michigan tributaries. Along the X, there's a bunch of fish species. Along the Y, there's the number of plastic particles that they found. And you see there's only one species where they found 0 plastic particles. And this is just one example of a whole bunch of different studies that have shown that microplastic particles are being ingested. If you could click again, please. I think we're going to the poll. So a question for you all is, if we know that microplastics are everywhere and we know they're being ingested, what does that really mean for aquatic organisms? And your options are A, it's bad, it's just plain bad. B, plastic is inert, it's totally fine. C, plastic is inert, but it may introduce known toxic chemicals into an organism. Or D, depends on the organism. Food web dynamics are complicated. And yeah, we're going for like the gut answer. So maybe however long you want to give it stuff.
Yeah, we'll just give it about 30 seconds to give people a chance. And also when the bars stop moving. Okay, we could probably go ahead. Thank you. So Karen, if you want to capture that, we'll go ahead and get going.
And we'll revisit this at the end. So, still moving. No.
Thank you.
Yep. Okay. All right, let me get, sometimes this slide doesn't like to advance. There you go.
Okay, so a couple of years ago, Thomas Hook, Tim Malinich, Zach Feiner, and myself, all of us were at Purdue University at the time, decided to conduct a meta-analysis and it was really driven by that question, like, is it a big deal if organisms consume microplastic? We looked specifically at consumption or feeding, growth, reproduction, and survival of aquatic organisms. Click. And so I wanted to back up just for a second and say, talk a little bit about what a meta-analysis is. So one thing you might think is it could be carefully, methodically searching through literature for pros or cons of exposure to microplastic. You might use Web of Science or Google Scholar as tools to conduct this search. If you could click, please. This is not a meta-analysis. A lot of people call this a systematic review. And even though you may have some clear steps that you follow, ultimately, if you're spinning it one way or another, the author's biases may affect what ultimately makes it into the final paper. If you could click again, please. Another thought is you could draw data from other analyses to examine trends. So say you look across a whole bunch of studies that looked at larval fish growth. They exposed fish to various concentrations of microplastics. You put all the data together, you plot them, you see what the trend is. This is totally made up. This is not real data. If you could click, please. That is also not a meta-analysis. This is an example of a secondary analysis, and that's because you're still using the data, like the raw data from the experiments themselves, and you're just kind of pulling it all together. It's not to say that a systematic review or a secondary analysis is necessarily a bad thing to do. It's just that it's not a meta-analysis. Click, please. So talking about meta-analyses, you have to start talking about effect sizes. So let's say these are some potential distributions of two different groups. Say the dark line is the control and the other one is the treatment. The means are a certain degree apart, which is shown by the arrows at the top, and the level of overlap is kind of circled at the bottom. If you click again, please. Here's another distribution, set of distributions where the means are the same distance apart. So you might think they're different, but if you click one more time, the area of overlap is a whole lot bigger, right? So there are a whole bunch of different ways to calculate effect size, but essentially what you're doing when you calculate effect size is you are incorporating both the measures of the means, but also a measure of the error or the distribution. So you're starting to take into account, okay, they are different, but how sure are we that it's a big difference? Where the one on the left, we might be more certain that there is an effect of the treatment versus the second one. Click again, please. So meta-analysis methods, you set your question and your criteria for whether or not you're going to include a study in your meta-analysis. You perform a search, and again, you would use like Google Scholar or Web of Science. You set those parameters beforehand, and then you extract from each thing the means, the error, the sample size usually, and then any qualitative or quantitative parameters that you're interested in, like taxa, location, concentrations that they were exposed to, a whole bunch of different options. And then you get a set of papers that have everything that you need. To up your sample size a bit, you can do what's called a forward and backward search. Is like you take those papers, you see who has cited them and who have they cited, and can we get any more papers that fit our criteria from those? Then you get your final data set. And we think meta-analyses are particularly useful for informing programming at Sea Grant because they're not like a one-off, right? It's not like in case X with species Y under conditions Z, Q, R, whatever, then microplastics are bad. It's looking a little bit more at trends. And because you're doing the effect size, you can sort of compare from completely different studies because you're sort of standardizing within their study and then compare. Click again, please. So back to our meta-analysis, we included papers that examined at least one effect of direct exposure to microplastics on fish or aquatic invertebrate consumption, growth, reproduction, or survival. They had to be an experiment, but we did allow field-based mesocosms if the papers were set up that way. And they also had to include a no microplastics control treatment. They also had to report means, sample size, and measures of variance for both the controls and the treatments. And you would be surprised how often people forget to say like what type of error they've returned. Anyway, click again, please. So here's an example of our results, and I'm happy to share the paper with anybody who wants to see it. I have a whole bunch of other resources too. But in the paper, you'll see things like this. So for consumption, along the X is different taxa, along the Y is the mean effect size, and then the black balls and the white dots, what happened is from certain papers, we could calculate more than one effect size, but then they're not truly independent of each other because they're from the same study. So we ran our analyses with every single effect size we could pull out. I think there were 375 of them. And then we ran our analyses again, choosing just one effect size per paper to see if things really changed. And then the sample sizes are above them. If they're starred, that means that the error bar isn't overlapping the zero, and it means that is like a significant effect. And you can see that if you lump all of the taxa together, then there is a significant effect of exposure to microplastic, no matter which effect sizes you use. That's the 94 and 27, those first two dots. But once you start to break things down a little bit and break things down by taxa, it gets a lot less clear. If you could click again, please. And so with growth, it's the same type of thing. When you loop everything together, there seem to be some negative impacts of exposure to microplastics. But once you start to break things down, everything's all over the place. Click again, please. And it's the same type of story with both reproduction and survival. So you can click one more time, please. So summary of our findings, and I'm happy to talk about these in more detail with anybody. Overall, we saw relatively few very clearly negative impacts of exposure to microplastics. We saw a whole lot of neutral impacts. That's when it's over. We saw no positive ones, but we saw a lot of neutral ones. In general, the trends that we saw were driven by zooplankton, which sort of makes sense if you think about the size of a microplastic particle versus something very small. It might be kind of a bigger deal if it gets in there. But some caveats are, there's potential for publication bias, which is the idea that if you don't have a strong result in the direction that everyone is expecting, people might not accept your publication for peer review. The sample sizes were wildly different. You may have noticed that on the things, but like, I think adult fish, there were only like 2 studies that we were able to find that fit our criteria. And this was like looking through like thousands of papers. They had to fit our criteria and they just didn't. And this was, we were doing the searches back in 2017. So the length of the experiments, very few of them were longer than like 5 days. The vast majority of them were like 2 days. And then the concentrations that they were being exposed to were generally far above what you actually see in nature. And so this isn't a knock on any of these. It makes a lot of sense that, oh, sorry, go ahead and click, please. And so our results are, that there are relatively few slam dunks that like, yes, microplastics are bad. Although if you look at our consumption results, it did seem to be that, feeding or consumption was affected by exposure to microplastics, which sort of makes sense. The microplastics get in there, food can't get in there. So things like that. And then zooplankton were more affected than other organisms. And if you think about like, okay, that's the base of the food web. If we're messing that up, that could potentially trickle up. That's one thought. But we also saw a lot of gaps. A lot of the studies were on Daphnia magna, which is a model organism for toxicological studies. There was a huge focus on beads and pellets rather than like microfibers or other types. And relatively few studies look at microplastics in combination with other stressors. And this is where I'm saying it's not a knock on this. It makes sense that you would have an organism that is typically studied for toxicology. It makes sense that you would have an organism that you can keep alive. It's just that at the time, the vast majority of the research that was being done was kind of in these little things. Since then, the paper had, like, people have been expanding. If you could click, please. So what others have found, go ahead and click. So this paper has been cited over 200 times. And if you look through them, and you can find a lot of good resources just by seeing who cited us, there's a lot really cool research that's recently coming out. But there are a couple of different trends. One is the idea that, plastic in oceans are mounting, but evidence on harm is surprisingly weak. There are a lot of people who are kind of trying to say, like, are microplastics really a big deal? Especially since we know there are other toxic contaminants out there that are actually definitely causing harm. Like, should we be spending so much time on microplastics? There are active debates about this. But I will say that, especially recently, there are a lot of colleagues there are a lot of people, John Scott is one team, but there are others who are looking at microplastics as a potential vector for chemical contaminants. And they actually did find that particularly plastics that were left out in the field and had like a biofilm, they could deliver things like Pfas to organisms. So go ahead and click again, please. And then just last week at the International Association for Great Lakes Research Conference, Sherry Mason, who is one of the world leading researchers on microplastics, she had this slide that was pointing out that there is more plastic lost to the environment than plastic that is being recycled. And then, in the end, she sort of said, it's not the plastic itself that is necessarily the problem. It's what is the plastic taking in? What if the plastic breaks down inside the organism? Those are more of the problem. So go ahead and click again, please. So if we go back to our poll, click one more time, please. I think C and D are probably the most correct, that maybe it's not the plastic itself that is the problem, but it may be bringing other stuff in. Food web dynamics are super complicated. People are learning more all the time. But I guess the one thing I would add is an option E from our study is that even if the plastic itself isn't a problem, if an organism's gut has a bunch of plastic in it, and then that organism is also experiencing thermal changes, habitat loss, other toxic pollutants, it's not going to help to have plastics there, I think. So click one more time, please. I think that's it. Yep, so I just wanted to acknowledge everybody that helped, especially the Purdue Interlibrary Loan Office. Go ahead and click again. I have a ton of resources that I'll be happy to share. And there's more too. There are a couple papers that just came out that are really cool and are probably relevant. Okay, thanks.
Thanks so much, Carolyn. And just to let everyone know, in addition to sharing the recording, we will also share the slides, if confirmed that that's okay, by the speakers afterwards. So, I mean, you can take a screenshot of this resources page, but it will also be, shared afterwards as well. And so does anyone have any questions for Carolyn? You can put it in the chat or the Q&A. We're watching both. So just
Stephanie, we have one question while we give people an opportunity to type any additional questions they might have. Carolyn, how many times did you say that the paper has been cited already?
It's 235, and which is crazy. But then so I am actually really, because the microplastics research has gone like whoop, and so I'm actually really excited to repeat it in a couple of years to see if some of those gaps can be filled in. So
Very exciting. Thank you.
sure. Okay. And if anybody has any questions, I tend to talk for hours about this, so I'd be happy to follow up with anyone. So
no. Great. No, Carolyn, yeah, I mean, congratulations. I think it's great the attention that paper has been getting, and thanks so much for sharing it with us today. I don't see any questions. If you think of one as we move on, please use the chat. We will be sharing or holding them. We have more time later on, but I'd like to now introduce our final speaker before the panel, who is Jamie DeWitt, is Associate Professor of Pharmacology and Toxicology at East Carolina University in North Carolina, and looking forward to hearing about her work on Pfas and Pfoa. So Jamie, take it away.
Well, thank you so much. And thank you for inviting kind of a non-Sea Grant person who's a laboratory toxicologist to talk about some of the research that we're doing here in North Carolina in my lab to address a contaminant of emerging concern that we call per and polyfluoroalkyl substances, or PFAS. And like Carolyn, I feel steeped in PFAS all the time. So if you could go to the next slide. So one of the things about PFAS that I think really has us concerned in the toxicology and ecological community is that the universe of PFAS is very, very large. Some estimates put the total number of PFAS at just under 5,000 individual compounds. Some estimates put the number at just under 10,000 individual compounds. And yes, PFAS are diverse in their structure, they're diverse in their chemistries, but they do have commonalities, and they do have common characteristics of concern that make some of us suggest that managing PFAS as a class may be one of the best options forward for protection of environmental health. Next slide, please. So now we have a poll, and this is to ask about your knowledge of PFAS. In what substances are they located? Where can you find PFAS in everyday items, for example? I'll just pause here or restart here. This is actually kind of a trick question because all of the substances listed there have had PFAS found within them or detected within them, from carpeting to dental floss to rain protective gear to nonstick cookware. And that's because of the products in which PFAS are used. Next slide, please.
Great, thanks. Karen, do you have it? And then move on.
Yes, thank you.
So there are no incorrect answers. Sadly. So as you can see from the figure here, PFAS are used in a huge variety of industrial processes, products, and consumer products. And they're largely used because of the strength of the carbon-fluorine bond. This is among the strongest bonds in chemistry. And so they're able to withstand all sorts of different conditions, from heat to chemistry to other types of situations where those properties are needed. Yes, they are used in materials that are essential for our health, especially during a pandemic. They're used to protect firefighters. They're used to protect healthcare workers because they provide water resistance and stain and oil repellency. But do we need for materials to just roll off of our carpeting if that means polluting the environment? So as we start to ask questions about why PFAS are problematic, we also need to ask questions about whether or not their chemistries are essential for various products and processes. Next slide, please. Some of the major environmental health concerns are associated with the EPBMT. So for those of you familiar with the PBT paradigm, I've added to it a little bit. We're concerned about PFAS because they can be emitted during production of products. So fluoropolymers, for example, are a type of plastic. In and of themselves, they are likely chemically inert, but some of the non-polymer PFAS are used to make the fluoropolymers. So there can be emissions of the PFAS that are concerning when those products are produced, perhaps during their use, and then at the end of their life cycle. So we need to consider a life cycle perspective when we talk about PFAS. The vast majority of PFAS are persistent. That means they are recalcitrant to environmental degradation. They don't get broken down by enzymes in our bodies. As far as we know, they're not easily broken down by microorganisms in the environment. In terms of human half-lives, they can range from days to years. And in the environment, we estimate that they can persist for perhaps centuries, which is an extremely long half-life compared to other types of persistent organic pollutants. They also can move from the environment into living organisms, and because of that long retention time in our bodies, they can bioaccumulate. They can also move to points distant from a point of emission. Some PFAS can travel up to 40 kilometers from a site of emission, and they're toxic. The vast majority of PFAS that have been studied for toxicity do produce adverse health outcomes, and I'll talk about those just a little bit. Next slide, please. One approach that we've taken here in North Carolina is to establish a network of university researchers with funding from our General Assembly to ask questions about what PFAS are in the environment in terms of in surface waters and in groundwaters. What PFAS are getting into people? How can we remove PFAS from our water environments? How do they move through the atmosphere? And then what do they do to organisms when organisms interact with them through the environment or through other types of products and processes? And on the next slide, I'd like to share a video with you that was made about the PFAS testing network.
Because PFAS include about 5,100 different chemicals, what we know about the effects of these chemicals on human health is limited to a fairly small number. We know a lot about one particular PFAS known as PFOA. Quite a suite of health effects have been associated with not only PFOA, but some of the other PFAS.
The challenge with these compounds is because of their properties of being persistent and being, water resistant, they're very long-lived compounds in the environment.
They've been found in air, they've been found in soil, sediment, water. And I think what's alarming to people is the issue of why these compounds are still in the environment, why they're still being used, why are we still getting exposed to them?
The PFAS issue has really raised an alarm about the most basic question, that is, can we drink our water? Anytime something this threatening erupts and information is lacking, it really sparks a lot of fear because we just don't know. And so I think the community has had a huge effect. There are true grassroots organizations that have popped up asking all the right questions and pressuring elected officials to find out the answers and do the right thing by this community.
So the PFAS testing network is a group of scientists from many different universities across the state, and we've been charged to help produce information about the extent of PFAS contamination, where it's coming from, where it's going, what it does to our health.
We're really fortunate to have some of the nation's leading PFAS researchers at these universities. And so we've been able to assemble a very strong team of researchers to address this question.
One of the great things about the PFAS testing network is that it brings together all these different ideas and all these different approaches so that we can better understand PFAS contamination across the state. It's a really amazing way to bring together faculty with different expertise to solve a problem.
All right, you can go to the next slide. I'm glad that I wore a different outfit today. I almost did wear the same outfit as in the video, and it was filmed several years ago. So in terms of health effects, we call PFAS multi-system toxicants. We now have evidence from epidemiological studies supported by toxicological studies in experimental organisms that PFAS can affect a range of systems in our bodies. I'm specifically interested in effects on the immune system as I'm an immunotoxicologist. So we're asking questions about how PFAS affect the body's ability to make antibodies against vaccines, for example. So if you go to the next slide, this is just a quick overview of the type of work that we're doing in my laboratory. So we're asking about how PFAS affect the adaptive immune system, which is the immune system that is so critical to you right now as we get through this pandemic and are vaccinated. And we want to have immune systems that protect us from the pathogens in the environment that may be having a negative impact on our health. We're trying to understand some of the mechanisms that are associated with how PFAS reduce the antibody response. And we're also charged with studying some of the PFAS that have been discovered here in North Carolina that, to our knowledge, have no toxicological data. So we're contributing novel information so that decision makers can make decisions to protect public health and environmental health from PFAS exposure. So if you go to the next slide, I think that's my last slide. And we have lots of funding in the laboratory right now to ask questions about PFAS. And that's why I said I was steeped in PFAS. That seems like everything we do right now is focused on PFAS, but with upwards of 10,000 individual compounds about which we have very little data for the vast majority, it's really critical that we direct research efforts towards PFAS and towards other compounds and towards things like microplastics, of which PFAS are also a group, so that we can make decisions about protecting the ecosystem and our health. So thank you.
Amy, thank you so much. And what a beautiful video. I like to have just kind of break up the presentation. Thank you so much. Well, Karen, do we have any questions?
Not at this time, Steph.
Oh, I see a couple in the chat. I think maybe, oh, Carolyn. So Carolyn had one. Are the suite of PFAS identified in your work similar to the suite found around the US, or is there some spatial difference?
So one of the approaches that our analytical chemists are using is a combination of targeted and non-targeted approaches. So they're actually evaluating a suite of 55 different PFAS, which is a little bit more than what are on the methods developed by the US EPA for PFAS in drinking water. So we have an expanded capacity, and we have identified some novel compounds. It's hard to say if these differ from what has been discovered in other parts of other parts of the United States, because not all states have the types of monitoring efforts we have. I was going to say Michigan is a state that has done a lot of monitoring, but they're using method 537, which again is a smaller set of analytes than what we're looking at here in North Carolina.
Thank you, Jamie. We have another question here from Jen. She wants to know, the network of scientists is wonderful, she states that, and then ask, how are you connecting with the state agencies?
So state agencies are part of our communication strategy. We have a data sharing team. That information is shared regularly with the state agencies. So we work very closely with the Department of Health and Human Services and Department of Environmental Quality. And two scientists, myself and Detlef Knappe from NC State, are on what's known as the Secretary Science Advisory Board. This is a body that's advisory to the DHHS and DEQ. So we have really good communication and fairly consistent communication. We're also funded through the General Assembly by an entity known as the North Carolina Policy Co-Laboratory. And this is an entity that regularly communicates with the General Assembly. And then that information also gets to our state level our state and federal stakeholder agencies.
Very interesting, thank you. Judy's asked, what is being done to determine the most cost-effective upgrades needed to treatment plants to enable them to extract more CECs?
I mean, I could tackle that. It's not really one of my areas. I know some of the approaches that are being done here in the state, but this sounds like it might be a question for somebody who knows more about water treatment in general.
Yeah, I don't know that any of our speakers kind of have the background or expertise to tackle that question. But I mean, I think it's a good one. I think that that's going to face treatment facilities around the country, so we can hold that one. We're recording questions that are coming in that we might not be able to answer directly today for follow-up after the webinar. So thank you for that.
Do we have any more time, Steph?
I think we have just like another minute for questions.
Okay, we have a good one that we'll throw out. Abigail has said that she first learned about CECs back in 2006. Why are these contaminants still called contaminants of emerging concern? Is there time for a new name, or do we need to think about the label of CECs are preventing us from communicating the most helpful messages to public and elected officials?
So I can, okay, go ahead.
Yeah. Mm-hmm.
Okay. I was just going to say, I think one of the things that's a challenge is the constant, there's always a new one, there's always a new one, there's always a new one. And I think there's a bit of a, there's a concern that, like, you know, everybody, it's the same with like invasive species, right? Everybody's shifting to the new one, but the other ones are still a problem. So I don't know if it's that there should also be raised awareness of the fact that, you know, these were emerging, but then there are some new ones that are emerging, but then there are also ones that are still a problem. Because I sort of feel like, and this is, I'm not an expert in this field, but I sort of feel like you hear about legacy contaminants and people focus a lot on that. You hear about contaminants of emerging concern, but there's a whole suite of contaminants in the middle. And I feel like they get sort of looped with like contaminants of emerging concern. I don't know what other people's thoughts are. I don't know that that helps, but I think it's just a thought.
Yeah. No, good. Thanks, Carolyn. And it actually sets up really nicely the panel, because that was one of the things that we had as a potential question for our panelists. So first, I want to thank all of the speakers that have gone so far. As we know, there is so much to learn and think about it when it comes to CECs. We knew it was going to be impossible to cover everything we wanted to touch on in today's webinar. So we wanted to end with a panel discussion that would provide some additional time for the speakers and other Sea Grant leaders in this area to reflect on their thoughts from where we go from here. And so we've invited 2 members of the Water Resources Visioning Team leadership to add additional regional diversity to the discussion, as well as some of the different background that they have on CEC work. So we're now going to be joined by Sam Chan, who is the statewide extension watershed health and aquatic invasive species specialist with Oregon Sea Grant, and Mary Donahue, program development and partnership specialist with Hawaii Sea Grant. So our approach to the panel will be somewhat informal. I'm going to kick off the discussion with a question that any of our speakers and panelists may answer. I have additional questions for the panelists as backups, but we've also gotten great questions for the panel from the audience already, and we encourage you to continue to put your questions in the chat in the Q&A box. So I'm going to stop the screen share because we don't have slides for this portion. And my first question to the panel is, in what ways do you think we can best leverage extension to help our communities, states, and regions address CECs? And Mary and Sam, since you're new and you haven't spoken yet, maybe you would like to kick us off?
Sure, Stephanie. Hello, everyone. And I want to echo Stephanie's thanks for joining us today, as well as the village that it took to put this together. And I would also like to encourage everyone who has any interest at all to join us in our water resources group and see what it's like. And we would certainly benefit from your perspective and look forward to that opportunity. So thank you for that. Before I start answering your question, Steph, I would like to say that I am using the term contaminants of ongoing concern when I refer to contaminants that may be our old tried and trusted contaminants, to delineate them. And I think it's also an opportunity with when we say contaminants of emerging concern, for example, with regard to plastics. We know they've been a problem since the 1960s, but as Carolyn pointed out, I like to describe the wild type plastic versus the laboratory plastic. And instead of those sterile polystyrene beads that they feed to copepods, which is a tremendous and extremely interesting experiment, the wild type plastic is, as she said, out in the world. It has its own microbiome that is dependent on the environment in which it is discarded or mismanaged, or its history as it comes from the watershed into the ocean. And the various chemicals that leach out and or accrete or absorb actually can present new challenges. And I think that very adequately fits into the challenges, contaminants of emerging concern category. And then the other thing I want to mention is that I heard someone earlier on ask about contaminant that was not included. And I also encourage us to have latitude when we consider what is included in contaminants of emerging concern. For example, microplastics are not a regulated pollutant at present, and yet they are a contaminant that we are interested in. So I think there's a lot of room for the work that we do, and I think there's a lot of great work already going on. And I know I kind of headed off in a different direction, Stephanie, but to circle back to your question about leveraging extension, I think that there are opportunities to work with what partners that maybe we don't work with as much. And I think the USGS Water Mission Area is a great example. They have the Water Resources Research Institutes, and some of our Sea Grant directors also serve as Water Institute directors. And there's also the USDA NEFA program, which has had some life breathed back into it with our new administration nationally. So I'll stop, and I want to turn it over to Sam, and then we can come back later if we want to for more comments. Thank you.
Thanks, Mary.
Well, first I want to say I'm not the expert on CECs. I think this whole group of almost 60 to 70 plus colleagues here, collectively, we can be very effective in terms of extension. So, where can Sea Grant be effective? Well, first, let me share with you some of the challenges. Oftentimes, when it comes to chemicals of ongoing concern or emerging concern, is that in our Sea Grant programs, we often fund research which is preliminary and looks more at perhaps the source and some of the fate and presence of these chemicals in organisms that are important to our Sea Grant programs. And this could be the seafood that we have, the animals and mammals that we work with, and of course our communities, people that live in areas that are further downstream in a watershed. And so the challenge is that if research determines that these chemicals are abundant in large numbers, as Steve and others have shared earlier, but yet the levels are low and we detect them in organisms that are part of our other extension efforts, like for example, oysters. How do we actually work with the stakeholders in sharing that information? Because just because we find, say, an emerging contaminant in, say, in shellfish, that does not mean that they actually have physiological impacts on organisms, as Carolyn had just shared. So how do we actually go about communicating that information is one of the challenges when we actually find that these chemicals are abundant and in diverse places and also in organisms that we value. And so the success that we found is to be upfront and straightforward, obviously share the limitations acknowledge all the many stakeholders that could be associated with those compounds, straight all the way up the whole stream. That's been one way, and also to acknowledge that that we're going to be continuing this work. So that's a challenge that, on one hand, that we all face in that, but then it's also an opportunity because then we can engage in that further dialogue. Is that and also thinking about further work into, so what does it really mean? Just because something's there does not necessarily mean an impact. So the other area in terms of success in extension is that extension folks are it's about engagement. It's about making connections. It's about the people we know. And so one of the areas that we can do is pull together the many different parties, as Mary Lee mentioned earlier, not just the federal and the state agencies, but also many of the entities that actually deal with processing water. Like, for example, the AGWA, which is the association, the Clean Water Associations. Ultimately, they have to treat that water. And also some of the producers that are involved with using these, in many cases, very important compounds, from pharmaceuticals to some of the plastics and more recently, some of the compounds that are used to minimize tire wear and the impacts that we're beginning to see on spawning salmon, for example. And so it's pulling together these different groups into these more integrative type of studies that look at the whole stream of contaminant, so that we realize that there's not necessarily one person who is part of the solution, but everyone realized that they have a stake in it. And so no one really is singled out. And so when Sea Grant funds some of the convening work, some of the early research work, by that convening and building trust within the stakeholders, we then are able to maybe be more competitive in larger proposals that we look at the system. And then finally, another success is really making those touch points or connections. I think one of our most successful programs that Michael mentioned earlier was us working with the American Veterinary Medicine Association. And when it comes to pharmaceuticals and personal care products, we all love our pets, and the more touch points we can have that, our pets need care products, they need medicines. And what am I doing with their care products and medicines? And then you connect it with yourself. Oh, I have elderly that I care for that need medicine. I use medications, I care products. So therefore, it becomes more a part of our life. And that's where thinking about reduction, that is, do I really need all these compounds? Maybe use what I need, and working, in this case, working with the veterinarians and prescribing really what's needed, that reduces what could potentially end up as waste stream. And then ultimately looking into the stewardship aspect, which is ways that we can dispose of compounds that we don't need anymore. And Michael and others mentioned these take back events. But when you actually have these touch points where you have pets and people, that's where some of our connection can be. So I'll move on. Thanks. Thanks for your time.
Great. Thanks, Pam. So then I'll just open it up to the rest of the panel. If any of the other speakers had thoughts on either the question or what Mary and Sam have shared so far.
I'll just piggyback off Sam if I could. I think really understanding the diversity of the community that our extension agents operate in is very helpful. This kind of goes, it sort of piggybacks off the CEC, but for fish consumption advisories, understanding how fish are consumed in different ways through different ethnic groups can be very important to help kind of reduce exposure for those populations. This was from Michigan Sea Grant, but they had, out of Detroit, they had a large Asian population that would use the fish in whole soup, versus kind of the materials that were being distributed, which only listed grilling or broiling the fish and allowing the fat to drip off. And in reality, that's not like broad spectrum. Like not everyone participate or does that sort of thing. So understanding our communities and knowing it, what those exposure pathways are to try to help mitigate exposure is, I think, also a very important aspect.
Great.
May I also add something?
Yeah, go ahead, June.
You know, again, I don't do outreach per se, but a lot of folks in the PFAS testing network do engagement, and I'm also doing some engaged research as well, where we interact with community members to answer questions, and they're active research participants. And this could range anywhere from citizen science to community members being full investigative partners in a grant. I'm actually working on a grant right now that will include a nonprofit community organization as with their leaders as principal investigators. So it's also a way to infuse change into communities, to get people to make changes or to speak out to get changes made at the state or local or federal level.
Great. Thanks. There's so many different aspects and pieces to this. Well, great. Well, we wanted to kind of build off of this discussion by giving our participants a chance to share kind of their experience in this area. So we're not doing a poll. This is going to be more like a low-tech Zoom. So we call it a chat storm. I'm going to ask a question and give everyone a few seconds, less than a minute, to type your answer. But don't press return until I tell you to. And then we'll kind of have this chat storm of answers through the Zoom, and then we'll capture that for some of our information and follow up after the webinar. So my question is for the Sea Grant people on the phone, if you've had success pursuing extension regarding contaminants of emerging concern. So don't have a lot of spots, so you'd have to explain briefly, but start typing and I'll tell you when to hit return. Okay. All right. Well, if everybody's ready, if you've had success and you want to hit return in the chat, so we can see what some examples might be. Great. So we have one related to microplastics and also engaging agencies and nonprofits. One person has more of a forward looking, they're hoping to engage soon, which actually is a nice transition into another question that I have for the panelists. Great. Well, thanks so much. So I know we're kind of getting close to the end of our webinar, but kind of with what we're seeing in the chat and some of the successes that people are having, but knowing that not all Sea Grant programs are currently working in this area, I think the last question for the panelists will be, how would an agent, an extension agent, or any person or Sea Grant program get started in this area if they don't have active programming? So from any of the speakers that have been involved, do you have any words of advice for those out there that might be new?
I'll share something. Most of us actually have NOAA labs in our state or within our region, and in our case here, our Pacific Northwest Fisheries Lab would really value more assistance when it comes to extension and outreach when it comes to their research, and so dealing with emerging contaminants. So that's one way. Reach out to your researchers at your local labs and at your universities and partner with them.
Stephanie, I would just add to SAM's, which I was going to tack on the Research University SAMs, so thank you for that, to also reach out to the Sea Grant Water Resources Visioning team and extension agents and faculty and specialists that might be on the team, and as we know, we have a wonderfully functional network that is very supportive, and information, studies, things like that we can share.
I'd like to add one more point is that normally take back events for pharmaceuticals are run by the DEA, and they're also co-hosted normally with colleges of medicine and colleges of pharmacy. But their tact is usually more on reducing the human toxicity aspects. And so when at Sea Grant, we can actually bring in the environmental and ecology aspects, it really broadens the appeal of these take back programs. So it's not just a DEA event, it's a sustainability and stewardship event, which we've had some success in.
Good. Anyone else?
I'll add one more point. The wastewater utilities are oftentimes looking for partners when it comes to outreach, because oftentimes they end up being the point source, or the ultimate source for not because they're direct source, but they're secondary, tertiary sources accumulations. So work with their wastewater utilities in forming these partnerships. They would really much value Sea Grant's extension and outreach angles.
Thanks. Thanks so much. I'm also looking back through the chat and the Q&A, and I think we might have missed a question from somebody, which might actually be a way to, and a nice way to kind of wrap it up. One more complicating factor, and this could be to any of our speakers or panelists. How do you feel about different state drinking water standards and levels regarding PFAS contamination? I think we've probably seen this with other types of contaminants as well.
I could answer specifically about PFAS. A really nice article came out in 2020 written by Gloria Post, who is a risk assessor in the state of New Jersey with their Department of Environmental Quality. And she emphasized that the differences among states aren't huge. It's not like a difference of 1,001. It's a difference of maybe an order of magnitude. But something that we've seen consistently happen is that values for MCLs and health advisories have continued to decrease as we get more information. So I'm hopeful that we'll have some responses at the federal level. If not, then I'm hopeful that other states will institute MCLs for PFAS.
Good. Thank you, Jamie. That sounds good. So Karen, I think that got all of our questions. Did we miss anything before
we do have a couple more.
Okay.
Sam had asked, what are some of the questions typically asked by policymakers on PFAS?
So in terms of the questions that I've seen at some of the meetings, they want to know about health effects. Why should we be concerned about PFAS? What do they do to our bodies? They also want to know about costs of removing PFAS from drinking water, which sometimes we can't really answer as scientists because we don't always understand the costs associated with remediation. They also want to ask about the technologies that are available for removing PFAS from drinking water, but health effects, costs, filtration.
Very interesting. Thank you very much, Jamie. We also had a question. Jennifer asked us, she said, now, in order to not scare consumers away from eating seafood due to so many health benefits from those the seafood dishes, how have we, or how might we encourage to compare contaminant levels in seafood versus store-bought food? That way to not really get people specifically switching from seafood to store-bought foods, but to kind of better judge between their options. Is that a challenge that we can, we've thought about, that we might be able to discuss and maybe find a way that we could do a better job of discussing these options with stakeholders?
I do know that there had been some comparisons, say, with beef and seafood. And so and this was beef for PCBs. and there's very similar levels. And so there, that goes on the same note as to try not to discourage people from consuming seafood. But I also think there is a very site-specific element to where locally, caught seafood is, harvested from, right? So if you're in an industrial area and you're in an old canal and you're catching seafood, might be different than if you were in a more remote area in the lake. I don't know how much scientific research has been done to support that type of study, but I know there have been efforts in terms of the Eat Fish Safe through Michigan Sea Grant. Really, it kind of leads with that messaging, right? So I'm not telling you to boom, fish consumption advisory, right? Here's how you can eat fish safely, because we don't want you to stop doing that. So I think messaging is very important, but then also figuring out those site-specific differences or making sure that consumers or anglers aren't going into restricted areas and consuming seafood from those.
I think another approach that we've actually funded a study on is to look at finfish species in the Gulf of Maine and look at contaminants. At the same time, look at omega-3 fatty acids and other beneficial components of the seafood. So it's not really comparing to land-based animal protein sources, but it's taking a more holistic view of seafood to give a balanced message about seafood is healthy. It does have some contaminants, but there's a balance there.
Good. Thanks. This is great. So I think we've touched on everybody's questions. We are kind of working to capture them all, and we'll follow up after the webinar if we do find some that we haven't answered. But I want to thank everyone again for participating in today's webinar. I hope you will all stay tuned for more information about the Water Resource Visioning Team's next webinar that will be later this summer, kind of August, September. We're planning on talking about stormwater, green infrastructure. Your feedback is important to us, and so we do have a survey, an evaluation survey that we are going to put in the chat box. It will also be included in a follow-up email that comes from Zoom after the event. So we encourage you all to fill that evaluation out and help us make improvements to our webinar series. And so if you're interested in learning more about the team or joining, please reach out to me at any time. And thanks again for joining us. We'll get the recording posted as soon as we can, and have a wonderful evening.